Cytoplasmatic accumulation may also be linked to proteins. Among the causes of protein accumulation, there is excessive reabsorption, a phenomenon that can be observed when glomerulus lose their selectivity, and consequently, the proximal convoluted tubule has to increase in reabsorption activity. Its cells have then to handle large amounts of proteins. However, this process is reversible. Overproduction of proteins, for example, occurs when immunoglobulins are produced in very large quantities by lymphoid tissue neoplasms. Another example is that of protein accumulation in the form of a knot of cytoskeletal proteins, especially neurofibrils. In fact, neurofibrils are apparently the underlying cause of neurodegenerative pathologies. Finally, protein misfolding leads to the formation of molecules that accumulate and take up space, thus engulfing the cell. But cells have refolding systems to correctly refold misfolded proteins. These systems may be of two types. One is represented by chaperonins, morphine molecules that, by using energy, are capable of correctly refolding misfolded proteins. Chaperonins may be constitutive and can be found in the endoplasmatic reticulum or in heat shock proteins, proteins expressly produced when proteins are supposedly misfolded. And in case of fever, for example, body temperature increases. Large amounts of heat shock proteins are produced to refold any protein with an incorrect form. Besides the chaperonin system, there is the ubiquitin proteasome system. Ubiquitin is a small molecule binding the proteins that have to be destroyed. Being complex, this bond firstly requires activation through an enzyme called E1, or ubiquitin activator hydrolysing ATP, which takes a pyrophosphate and uses it to bind ubiquitin and transfer it on E2, a ubiquitin carrier enzyme, whose role is to bind E3 enzyme or ubiquitin ligase. Ubiquitin ligase then transfers ubiquitin from E2 to the substrate. Every substrate marked with 4 ubiquitin proteins is considered a target by the proteasome, an enzymatic complex that destroys the target molecules. These two systems are enough to block excessive protein misfolding. In fact, in this case, unfolded protein response can be observed. Unfolded protein response is the gene activation inducing an enormous amount of heat shock protein production. However, when this response isn't enough, proteins accumulate in the endoplasmatic reticulum. When this mechanism is over, the cell cannot be saved and therefore enters via the intrinsic pathway apoptosis. A very particular example of extracellular, not cytoplasmatic accumulation is that of amyloids. Amyloidosis is the extracellular buildup of amyloids and is a frequent cause of many diseases. It is related to the accumulation of amyloid, a protein that is not chemically well defined. However, the term is generally used to indicate a protein with beta-pleated sheet that tends to accumulate extracellularly. A characteristic feature of amyloid is Congo red positivity and birefringence in polarized light. Amyloid is a protein with an antiparallel B-pleated sheet with possible presence of proteoglycans, glycosaminoglycans, and serum amyloid P component inside. Amyloid is usually a physiological protein that somehow accumulates. But there are different forms of amyloidosis. The first one is AL amyloidosis, where amyloid is the immunoglobulin light chain. This phenomenon can be observed especially in multiple myeloma, a tumoral form where plasma cells expand and start to produce so much immunoglobulin that it accumulates everywhere in the cell, even inside the nucleus. These cells usually produce immunoglobulins with some defects, or sometimes only light chains leading to Banks-Jones proteinuria. Another form of amyloidosis is AA amyloidosis, where serum amyloid, an apolipoprotein that is synthesized in the liver and works as an acute phase protein, accumulates. A beta amyloid is a totally different thing. It is typical of Alzheimer's disease, the most frequent cause of dementia in old people, which affects up to 40% of the population aged 85 to 89. Its etiology is linked to the cleavage the serum precursor protein of beta amyloid undergoes inside the membrane of the neurons. In healthy patients, alpha and gamma secretase cut beta amyloid, leading to soluble fragments. In patients with Alzheimer's disease due to genetic or acquired causes, secretase do not carry out their function correctly, so beta amyloid isn't cut and accumulates. From a histopathological point of view, there is a loss of cerebral tissue with neuritic plaques until complete destruction of cerebral tissue. ATTR amyloidosis is linked to transthyretin, a plasmatic protein that normally carries thyroxine and retinol. This form is also called senile systemic amyloidosis and is a familial form where accumulation occurs mainly in the cardiac system. 2 beta amyloid is related to the accumulation of beta-2 microglobulin, a part of MHC of class 1 molecule, which was not filtered by the old dialysis machines and therefore circulated in the body and accumulated even more over time. Today, hemodialysis is more modern and has sorted out this problem thanks to filters that are capable of getting rid of beta-2 microglobulin. Finally, there is amyloid caused by prions. Prions are extremely strange pathogens since they are proteins capable of duplicating themselves, inducing conformational change into similar proteins. Prion protein accumulation is the underlying cause of bovine spongiform encephalopathy. Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.